Good afternoon. I am Eugen Chycewicz and I'm co-founder and CEO of Medicem. Medicem is an integrated group of biomedical companies that is developing innovative medical devices, and we are focusing on ophthalmology. Our core scientific competence lies in bioanalogic hydrogels, the hydrogels that are resembling very closely those that are appearing in the human body. We building on the heritage of Professor Otto Wichterle, who is credited as the father of soft contact lenses, and we are further building the portfolio of scientific knowledge based on bioanalogic designs of intraocular lenses, and we have more than 20 years of experience in that area. In the previous slide, I have used pretty often the word bioanalogic, so probably it would be good to explain what we mean by that. Basically, applying a bioanalogic approach means that we would look at the organ that we would like to replace with an artificial implant, and we identify what are the critical features of this organ. And then we try to create something like a functional analogy, not necessarily a copy of that organ, let's say, built out of artificial materials. What would that mean, for instance, in presbyopia surgery and, let's say, presbyopic implants? When we look at presbyopia correcting intraocular lenses today, there is a lot of limitations of existing technologies, especially when we compare them with natural young crystalline lens. We can learn a lot from human eye, how human eye is making sure that we see on all distances, that we see in all light conditions. We also see, and we learn from human eyes, that natural crystalline lenses are very variable, that they are changing in time, they are featuring very variable optics, they have very poor MTF or other lab parameters we can usually measure on the optical bench. And probably natural crystalline lens would never be going through a regulatory scrutiny and never get a regulatory approval. On contrary to that, human natural crystalline lens is still the best for our vision. Why is that? We believe that the answer is that our vision is not only driven by optical characteristics of the lens, but it is to high extent determined also by the quality of signal processing in retina and brain. This bioanalogic approach to intraocular lens design leads us to defining a new paradigm. So instead of searching for an optically perfect lens, let's try to develop the one for maximum optical quality, the one that is most similar to the natural crystalline lens, that features very similar ways of ensuring vision on all distances at all light conditions, ensuring the similar ways of signal processing in retina and brain. In Medicem, we have designed such first bioanalogic intraocular lens. We call it WIOL-CF. And on this slide, I present the key features of the lens. The first key feature is hyperbolic curvatures of the surfaces, which are leading to an optic with negative spherical aberration, which is natural to human eye, and that is providing large depth of focus, which is one of the mechanisms to ensure vision on all distances. Also in hyperbolic optics, you have most of the optical power in the middle, in the center of the lens, which is then, let's say, a prerequisite for pseudo-accommodation to work, which is another mechanism to ensure vision on all distances. Our lens also is characterized by full 9mm optics, which is allowing a lot of light getting in the eye, which is then helping a lot in mesopic contrast sensitivity. In material, we've been also following the bioanalogic principle, and we have made the material to resemble the most the natural crystalline lens material. This material is featuring negative charge, which is helping for long-term biocompatibility, because the lens is supposed to stay, say, for a very long time in the eye, and let's say we are expecting long-term function. And also we've been trying to achieve a simple design that would enable broad use in routine cataract surgery. On this slide, I'm trying to match our solution with common problems in cataract or presbyopic surgery and compare it also with the other approaches that are used in this area. So when we look at vision on all distances, we see that there are multiple solutions being deployed in the past years. WIOL-CF attempts this via depth of focus and pseudo-accommodation, and you see that two of these mechanisms are also natural to natural crystalline lens. Optical phenomena, WIOL-CF is built from the material with refractive index which is similar and the same as the natural crystalline lens. This is one of the ways how to minimize optical phenomena. It has no optical zones, so no reflections created on those. No haptics, it has full optics, so again, no reflections created on those. With regards to contrast sensitivity, again, the full optics is helping WIOL-CF to reach very good levels. No diffractive and refractive zones are again very helpful. And for the long-term and stable function, we created this lens from highly biocompatible material that is featuring negative charge, that is discouraging proteins or cells from adherence and discouraging any cell growth. So all that is theory, but what is important is also practice. On this slide, we summarize our current clinical trials. And most of our clinical data is coming from a multicenter trial that was run in 2013 in the Czech Republic. Here I present the results from 12 centers, 62 patients, 124 eyes with 12 months follow-up. As you can see, we have achieved a very good, excellent result on distance, excellent result on intermediate, and good result on near vision. What is even more important is that we have realized some further areas of optimization that are listed in the table below. At the time of the Czech trial, we only had available power lens calculation by SRKT formula, which we've been optimizing for the further trials. We've been, further working on optimization of binocular vision, and we also realized how important is centrally placed rounded circular capsular axis, which is enabling good lens position without haptics. So implementing all of these opportunities for improvement in the subsequent trials, you can see that in the single center trials with Dr. Bobby Young, we achieved comparable results on distance, but we improved intermediate and near vision, and the same we continued in the other single center trials at Dr. Daya Center in UK and Dr. Mertens in Belgium. In all of these trials, we have consistently found excellent mesopic contrast sensitivity that was exceeding healthy population norm and glares and halos levels at the level of monofocal intraocular lenses. Based on these very encouraging single center trial results, we have agreed with Bausch & Lomb to jointly set up, fund, and execute a broader European multi-center clinical study to validate this pilot study's results and also to define and validate the optimum clinical procedure for WIOL-CF. And this clinical procedure would be involving femtocataract laser surgery. We were giving the trial acronym BOND for this study, which means uniting bioanalogic lens and femtosecond laser surgery. We plan to recruit 150 bilaterally implanted patients with cataract and presbyopia. We have actually already started the trial. We have first patients recruited in. The patients will be followed up for 6 months, and we are having clinical investigational centers in most of the countries of Europe. In summary, bioanalogic hydrogels present numerous properties that make them one of the most suitable materials in ophthalmic surgery. In combination with bioanalogic designs, they are facilitating the applications and outcomes that are impossible with existing technologies. They can do so not only for presbyopia or cataract surgery, but they can also be developed in the applications for glaucoma, vitreous body replacement, phakic IOLs, corneal inlays, or laser adjustable intraocular lenses. We understand WIOL-CF as being the first proof of concept of bioanalogic design for intraocular lenses, and we are also developing follow-up products to WIOL-CF. Thanks to the combination of material and design features, bioanalogic design features. WIOL-CF itself brings back the characteristics of natural vision. And we are now, as a company, focusing at maximum at EU clinical trial in collaboration with Bausch + Lomb that is underway to validate highly encouraging results of single-center trials. Thank you for your attention.